Good morning, guys. So today I'll be talking about apoptosis. So if you see, basically apoptosis is an important topic, be it in UG level or in entrance examination point of view. So we'll be talking about apoptosis in detail, like the mechanisms and the causes of apoptosis, like what are the different causes of apoptosis, and I will also be trying to explain the format in which we can write during our university exams, right? So basically, if we start apoptosis, we'll start with the definition, right? So this is the definition that is given in Robbins. So it basically says that apoptosis is a type of cell death that is induced by tightly regulated. It's actually a suicidal program, right? So it's a tightly regulated suicidal program in which the cells which are destined to die, they will activate certain intrinsic enzymes that will basically degrade the cellular DNA and nuclear and cytoplasmic proteins, right? So these are the two key points which you should remember. See, if if the cellular DNA and the nuclear and cytoplasmic proteins are gone, like they are damaged or if they are degraded, then the entire cell should definitely die. So it is a regulated program, right? It's a regulated suicidal program. So it's also a key point that you should remember, right? So this is about the definition. Now let us talk about the causes of apoptosis. So there are basically two causes: physiological cause and pathological cause, right? So if you see the physiological cause, now there should be purpose for both these causes, right? So we'll see the purpose for physiological cause. So in physiological cause is basically natural phenomena, and here we need to eliminate the cells that are not needed, right? So there is the elimination of the cells that are not needed, and we need to also maintain the constant number of cell population. So these are the three important reasons for which there is apoptosis that occurs physiologically, right? So we'll talk about them in detail. So if you see, in the first case we have the removal of supernumerary cells. So what do you mean by supernumerary cells? So basically, when there is, see, in a particular type organism, there should be a particular number of cells. So when they exceeding, like when there's more number of cells, we refer that to as supernumerary cells. So there can be two things. One is apoptosis. During apoptosis, cell death during involution of primordial cells and remodeling of maturing tissues. So what do you mean by involution? Involution basically refers to shrinkage of the primordial cells. If you have learned during that in the embryology, you'll be probably knowing that during embryology there are certain cells that are involuted, right? There are shrinkage of certain cells and there is remodeling of the maturing tissues which occurs embryologically. So that during those stages also we require apoptosis. And another thing is that apoptosis during development is basically called programmed cell death, right? So basically during development, whenever apoptosis occurs, we refer that to as programmed cell death. However, the involution of the primordial cells and rematuring of the remodeling of the maturing tissues can also be called as apoptosis. So this comes under removal of supernumerary cells. Secondly, we have involution of hormone dependent tissue on hormone withdrawal. So basically this refers to the apoptosis that occurs in a particular tissue whenever there is deficiency of a particular hormone. So if you see, basically during menstrual cycle we require the hormone called progesterone to maintain the endometrial lining. So whenever there is deficiency of this progesterone, okay, so what will happen? There will be endometrial breakdown. So that is the thing we have written here: endometrial breakdown during menstrual cycle due to the deficiency of progesterone. We also have an hormone called FSH and LH. So they are basically responsible for the maturation of the ovarian follicles. So after menopause, there will be decrease in the FSH and LH levels. So during that time, there will be ovarian follicular atresia in menopause. We also have a hormone called prolactin. We know that prolactin is responsible for secretion of the milk. So whenever there is regression of the lactating breast whenever there is weaning, like after we stop giving breast milk to the young child, what will happen? There will be regression of the lactating breast because there is no formation of that particular hormone called prolactin. Right. Now we'll talk about the third point. So third, we have the elimination of the lymphocyte that fail to produce functional antigen receptors. So basically, immature lymphocytes in bone marrow and thymus. There will be some immature lymphocytes in the bone marrow and the thymus, and even the germinal center B cells. So we need to get rid of these immature lymphocytes. So that is why there is elimination of the lymphocytes, right? Next, we have elimination of potentially harmful self-reactive lymphocytes. Right. In the fifth point, we have the death of neutrophils and lymphocytes. So basically, neutrophils serve their purpose of acute inflammatory responses, and lymphocytes at the end of immune responses. So after this particular immune responses or inflammatory responses, we no longer need their presence, right? So there will be death of these neutrophils and lymphocytes. So that's another cause of physiological apoptosis, right? Now we'll talk about pathological cause. So in pathological, what is the purpose for pathological causes? We need to eliminate the cells that are injured beyond repair. So that, see, basically whenever there is damage to a cell, there will be certain repair mechanisms, okay, which will cause the cell to bring back to normal. But whenever there is some repair whenever there is damage that occurs to the cell which cannot be repaired, so then there is no need for the cell to be present in the cell, right? So if you talk about the causes, first one we have the DNA damage. So whenever there is radiation or any drugs, okay, that can cause the DNA damage, free radicals will be produced obviously, and these free radicals, okay, will cause the repair mechanism to fail, and this will result in apoptosis. So basically the damage is done beyond repair, so beyond which the repair mechanism cannot work properly, so apoptosis occurs. So that is DNA damage. Second, we have accumulation of misfolded proteins. So whenever there is improperly folded intracellular protein, that can lead to cell death. Third, we have apoptosis induced during infections. We know that during infections, right, so there will be see, if you can see here, there is an infected cell, okay, probably due to a virus, and the body tries to produce T lymphocytes, the cytoplasmic T lymphocytes, against this particular virus. So what will happen? There will be apoptosis, right? We already learnt about the T lymphocytes, cell-mediated immunity and all. So in which the T lymphocytes were produced in excess, and these caused the apoptosis of the infected cell. Now we'll talk about the fourth cause, which is the pathological atrophy in parenchymal organs after duct obstruction. So basically what happens is, whenever there is a duct, if you imagine this to be a duct, right, so whenever there is some duct obstruction, if there is an obstruction over this part, what will happen is there will be some pressure that is built up over in this particular area, right, like beyond the, like before the point of obstruction. So what will happen, there will be certain, because of the pressure, what will happen is there will be atrophy of that parenchymal cells that are present over this region. So this can occur in the organs like kidney, pancreas and parotid gland. So that is the fourth point. So these are the physiological and pathological causes of apoptosis, right? Now we'll talk about the mechanism of apoptosis. So basically there are two important mechanisms. One is an intrinsic pathway, another is an extrinsic pathway. In intrinsic pathway, it is also referred to as a mitochondrial pathway, and extrinsic pathway is also referred to as death receptor initiated pathway. Okay, so we'll talk about intrinsic pathway first. So before we start the intrinsic pathway, we need to remember three important points. One is called anti-apoptotic proteins, another is a pro-apoptotic protein, another is a regulated apoptotic initiators. So what is an anti-apoptotic protein? Basically there are three important things. See, there are many other important proteins, but we'll remember the commonest ones. So first one is the BCL2, BCLXL and MCL1. If you see in anti-apoptotic proteins, we have 4 BH domains. So they can be either BH1, BH2, BH3 or BH4, right? So these are the 4 important BH domains. So they are present in anti-apoptotic gene. So they are present mainly in the outer mitochondrial membrane of the cytosol and endoplasmic reticulum, right? So, and they are also responsible for making the outer mitochondrial membrane to be impermeable. So they basically this basically prevents the death-inducing proteins like cytochrome C into the cytosol. So what they are trying to say is basically simple. So if you take this to be a mitochondria, right? So this is the outer membrane, this is the inner membrane, right? So let me try it more perfectly. So this is a mitochondria, right? So this is the outer membrane and this is the inner membrane. So these anti-apoptotic proteins are basically present in the outer mitochondrial membrane, and they make the outer mitochondrial membrane more impermeable to what? To basically the the cytochrome C and the apoptotic inducing proteins or death inducing proteins. Okay, so one of the example is cytochrome C, right? So that is about anti-apoptotic protein. Now what about pro-apoptotic proteins? So what are the examples? Some of the examples are BAX and BAC, right? Let me clear this off. So some of the examples are BAX and BAC. So there are basically 3 BH domains, that is BH1, BH2, BH3. So they act opposite to that of anti-apoptotic proteins. They basically enhance the outer mitochondrial membrane permeability. So probably by forming a channel in the outer mitochondrial membrane, right? So there is no specific demarcation, like there is no specific indication for the formation of this particular channel in the outer mitochondrial membrane, but we mostly take it as by forming an channel in the outer mitochondrial membrane. So basically it acts like this, right? So by this channel they basically push out the cytochrome C or the other death inducing proteins into the cytoplasm. So that is about pro-apoptotic proteins, right? So we need to clearly understand that pro-apoptotic proteins will enhance the permeability of the outer mitochondrial membrane to the death inducing proteins, whereas anti-apoptotic proteins will inhibit or will prevent the permeability of the outer mitochondrial membrane and prevent the entry of the death inducing proteins into the cytoplasm. Now we'll talk about the regulated apoptotic initiators. So basically they are also known as the sensors of cellular stress. So when they are upregulated, apoptosis is induced. Some of the examples include BIM, BID, BAD, PUMA and NOXA. So they basically contain only one BH domain, that is BH3. Right. So they contain 4 BH domain, they contain 3 BH domain and they contain 1 BH domain, that is BH3. Right. Now go in detail about intrinsic pathway. So what happens in intrinsic pathway basically? So this is the flow chart that is also given in Robbins. Let me try to make it more simple and put it in simple terms. So first there should be stimulus, right? So what is the stimulus for apoptosis? First is the growth factor withdrawal, absence of survival signal. See, this also refers to that whenever there is no survival signal, like whenever there is no signal that the cell should survive, then there will be apoptosis, right? And whenever there is any protein misfolding or whenever there is a DNA damage. So these all 4 causes are the inducing point for apoptosis to occur, right? So I already talked about the misfolding, protein misfolding and DNA damage in the physiological causes, and these are all of the physiological causes basically. So what happens is whenever these particular whenever these 4 or the death inducing situations are induced, what will happen is the BH3 receptor is upregulated. I already told that BH3 sensor is actually what? It's actually a stress sensor. I already mentioned over here, right? Sensors of cellular stress. So these are the regulated apoptotic initiators. Let me clear this off. These are basically regulated apoptotic initiators. So whenever the BH3 sensor is upregulated, what will happen is the backs and back. So what are backs and back? You can see over here, right? So they are pro-apoptotic genes. Backs and back are pro-apoptotic genes. So these pro-apoptotic genes are activated. So they basically come under BHC2 family effectors. So whenever they are activated, what will happen? The backs and back will dimerize to form a channel, and I told you about this particular channel, right? You can see over here. So these channels are responsible for traveling or traveling the cytochrome C from the inner mitochondrial membrane into the cytoplasm. So we already know the cytochrome C is also present in the ETC. So basically in Robbins they would have told about cytochrome C as a double-edged sword. Basically, what is the meaning of that is, see, we already know cytochrome C is responsible for production of ATP during ETC. That is a positive sign for that. So it makes the cell alive. Whereas in this particular apoptosis, cytochrome C causes the apoptosis, right? So it's a negative sign, or it makes the cell to die. So that's why it's a double-edged sword. Robbins has mentioned that in his book. Now we'll talk about it further. So now what happens is cytochrome C and other pro-apoptotic proteins. It need not be only cytochrome C, there can be other pro-apoptotic proteins as well. So what will they cause? What will they do? They will come and attach with APAF, apoptosis activating factor 1. Now when they come and attach, they form a structure called apoptosome. This apoptosome will cause the activation of procaspase 9 into caspase 9. Now we have a protein called caspase 9, right? Now caspase 9 is activated. So the main function or the main motive of intrinsic pathway is to produce caspase 9. So we have produced that. Now there are some other mitochondrial membranes that enter the cytoplasm from the mitochondria. So they are called SMAC and Diablo. So they basically bind and neutralize IAPS. What do you mean by IAPS? Is inhibitors of apoptosis. Whenever the inhibitors of apoptosis are neutralized, what will happen? Apoptosis is initiated. So caspase cascade pathway is initiated. So these are initiated by the SMAC and Diablo. These are the other mitochondrial membranes present in the mitochondria. So this is the overview of intrinsic pathway. Now let us go to extrinsic pathway. So in the extrinsic pathway, let me talk about the structures that are present. Firstly, we have a substructure called FAS receptor or the TNF type 1 receptors. They are also called CD95. We also have a structure called death domain. See, all the TNF receptors need not be associated with a death domain. Whenever they are associated with a death domain, then they are involved in apoptosis. So if they are not associated with a death domain, then they are basically involved in inflammatory cascades, right? Now, let me talk more about the structure. So firstly I told about the FAS receptor, and I told about the death domain, and next we have something called as the FAS ligand. So whenever the FAS receptor binds to FAS ligand, then there is the extrinsic phase that's activated basically. So now when the FAS ligand, see, you can see over here, I can see over here, the FAS ligand is attached to the FAS receptor. So what happens is the FAS associated that domain is formed. So basically what happens, FAS receptor is bound with FAS ligand. Let me clear this off. FAS ligand, this produces FADD. What do you mean by FADD? Is FAS associated death domain. So what does this FADD does is, it basically activates the procaspase 8 or 10 into caspase 8 or 10. Basically converts the procaspase into active caspase. So there's something called the flip protein, which when attached to this procaspase 8 or 10 will inhibit FADD and thereby inhibiting extrinsic phase, right? So we talked about the extrinsic phase. Now there's some interaction between the extrinsic phase and intrinsic phase. Basically, if you see in hepatocytes and pancreatic beta cells of pancreas, in hepatocytes and beta cells of pancreas, the caspase 8, I told you the caspase 8 is formed in extrinsic pathway. It basically activates BID. Where did you see this particular the enzyme called BID? We saw this in sensors of cellular stress. So basically, when they are when the sensors of cellular stress are upregulated, what will happen? Apoptosis is induced. So caspase 8 activates BID, right? Now now this is referred to as the interaction between the extrinsic and intrinsic phase. This is also a small concept that is given in Robbins, right? Now let us talk about the execution phase. Let me clear this off. Right, what happens in execution phase? See, I told you see, what happens in execution phase is that there's something called as caspase 9. We told that caspase 9 is formed in intrinsic pathway. In extrinsic pathway, we have the caspase 8 or 10 that is produced. So whenever caspase 9 and caspase 8 or 10 combine together, they activate the caspase 3 and 6. So this function of caspase 3 and 6 is to cleave the inhibitors of DNase. So basically DNase is an enzyme that degrades the DNA. When the inhibitor of DNase is inhibited, what happens? When the inhibitor of DNase is cleaved, what will happen? The nuclear fragmentation occurs. So DNA fragmentation occurs. So that's the thing that occurs in execution phase. So another thing that occurs is that in normal healthy cell, there will be something known as the phosphatidylserine, which is present towards the cytoplasm, right, towards the cytosol. So in apoptotic cell, there is a flipping that of the phosphatidylserine that takes place, and this type of structure will be formed that's mostly present in the apoptotic cell. Basically, there's flipping of this phosphatidylserine, right? Now what happens in this apoptotic cell further is that, so basically you can see over here some proteins or ligands that are present over the surface of the apoptotic cell, right? These proteins or ligands act as an eat me signal. So what do you mean by this eat me signal is that whenever they are present over the surface, they give a signal to the macrophages or the phagocytes to come and engulf them. So they also secrete certain other proteins, okay, which will attract the macrophages towards them and cause them to engulf. So this type of phagocytosis, right, is known as efferocytosis. Okay. Now, apoptotic cell can also become coated with natural antibodies and proteins of complementary system like CLQ. Right. Now, what happens after the ingestion of the apoptotic cell? There is basically production of pro-inflammatory cytokines in the macrophages is reduced. So basically, whenever the macrophage engulfs something, what will happen is there will be inflammatory cytokines that are produced. But in this apoptotic cell, whenever there is ingestion of the apoptotic cell, there will be production of pro-inflammatory cytokines which are reduced in macrophages. Normally, the macrophages will produce these cytokines, this inflammatory cytokines, pro-inflammatory cytokines. But in an apoptotic cell, when it is ingested, the pro-inflammatory cytokines production is reduced. So because of which inflammatory reactions are reduced in apoptosis. Whereas in necrosis, if you remember, there will be inflammation that occurs. So in my next video or in my further videos, I'll be talking about the differences between the necrosis and apoptosis. during which you will understand why there is no inflammation in why is there inflammation in necrosis. However, now you understood why there is no inflammation in apoptosis, right? So this is all about apoptosis. I guess you understood the pathway as well as the causes and the simple definition about apoptosis. Thank you for watching.